The overall goal of this procedure is to investigate the cellular and molecular mechanisms of neotissue formation and stenosis development in tissue-engineered vascular grafts. First, biodegradable tubular scaffolds are assembled by coating polyglycolic acid nonwoven felt mesh with an epsilon caprolactone and L-lactide copolymer. The next steps are to harvest bone marrow and isolate the mononuclear cells by density gradient centrifugation. Then, approximately 1 million cells are seeded on the scaffold, and they are incubated overnight. The final step is to implant the seeded scaffold as an inferior vena cava interposition graft. Ultimately, results can show cell infiltration and extracellular matrix deposition in the tissue-engineered vascular graft through immunohistochemistry.
The implications for this technique are to better understand what causes the formation of stenosis in tissue-engineered vascular grafts, and then use this information to design a better tissue-engineered vascular graft. From our clinical study, we learned that nearly a quarter of patients who received the tissue-engineered conduit developed a narrowing. Our goal is to eliminate this problem.
Although this method can apply to the congenital heart disease, it also can be applied to the coronary bypass surgery or the conduit for the arterial venous fistula.
Generally, individuals new to this method will struggle because of the fine microsurgery techniques required during the vein osmosis.
We first had the idea for this method when we were looking for a better way to characterize the new tissue formation and ways